Forensic entomology, Wikipedia audio. Forensic entomology is the scientific study of the invasion of the succession pattern of arthropods with their developmental stages of different species found on the decomposed cadavers during legal investigations. It is the application and study of insect and other arthropod biology to criminal matters. It also involves the application of the study of arthropods, including insects, arachnids, centipedes, millipedes, and crustaceans to criminal or legal cases. It is primarily associated with death investigations. However, it may also be used to detect drugs and poisons, determine the location of an incident, and find the presence and time of the infliction of wounds. Forensic entomology can be divided into three subfields: urban, stored product, and medico-legal/medico-criminal entomology. Historically, there have been several accounts of applications for, and experimentation with, forensic entomology. The concept of forensic entomology dates back to at least the 13th century. However, only in the last 30 years has forensic entomology been systematically explored as a feasible source for evidence in criminal investigations. Through their own experiments and interest in arthropods and death, Sung Tzu, Francesco Redi, Birgit Darbois, Jean-Pierre Megnin and the physiologist Hermann Reinhardt have helped to lay the foundations for today's modern forensic entomology. The Song Dynasty Forensic Science book collected cases of injustice rectified, published by court judge, physician, medical scientist, and writer Song Ci in 1247 contains the oldest known case of forensic entomology. In a murder case of 1235, a villager was stabbed to death and authorities determined that his wounds were inflicted by a sickle. This was a tool used for cutting rice at harvest time, a fact which led them to suspect a fellow peasant worker was involved. The local magistrate had the villagers assemble in the town square where they would temporarily relinquish their sickles. Within minutes, a mass of blowflies gathered around one sickle and none other, attracted to the scent of traces of blood unseen by the naked eye. It became apparent to all that the owner of that sickle was the culprit, the latter pleading for mercy as he was detained by authorities. History. Song Ci was a judicial intendant who lived in China 1188 to 1251 AD. In 1247 AD, Song Ci wrote a book entitled Washing Away of Wrongs as a handbook for coroners. In this book, Song Ci depicts several cases in which he took notes on how a person died and elaborates on probable causes. He explains in detail on how to examine a corpse both before and after burial. He also explains the process of how to determine a probable cause of death. The main purpose of this book was to be used as a guide for other investigators so they could assess the scene of the crime effectively. His level of detail in explaining what he observed in all his cases laid down the fundamentals for modern forensic entomologists and is the first recorded account in history of someone using forensic entomology for judicial means. In 1668, Italian physician Francesco Redi disproved the theory of spontaneous generation. The accepted theory of Redi's day claimed that maggots developed spontaneously from rotting meat. In an experiment, he used samples of rotting meat that were either fully exposed to the air, partially exposed to the air, or not exposed to air at all. Redi showed that both fully and partially exposed rotting meat developed fly maggots, whereas rotting meat that was not exposed to air did not develop maggots. This discovery completely changed the way people viewed the decomposition of organisms and prompted further investigations into insect life cycles and into entomology in general. Blowflies family Caliphoridae. Flies in this family are often metallic in appearance and between 10 and 14 mm in length. In addition to the name blowfly, some members of this family are known as blue bottlefly, cluster fly, green bottles, or black blowfly. A characteristic of the blowfly is its three-segmented antennae. Hatching from an egg to the first larval stage takes from 8 hours to 1 day. Larvae have 3 stages of development, each stage is separated by a molting event. Worldwide, there are 1,100 known species of blowflies, with 228 species in the neotropics, and a large number of species in Africa and southern Europe. The most common area to find Caliphoridae species are in the countries of India, Japan, Central America, and in the southern United States. The typical habitat for blowflies are temperate to tropical areas that provide a layer of loose, damp soil and litter where larvae may thrive and pupate. The forensic importance of this fly is that it is the first insect to come in contact with carrion because they have the ability to smell death from up to 10 miles away. Dr. Louis-François Bergeret was a French hospital physician, and was the first to apply forensic entomology to a case. In a case report published in 1855 he stated a general life cycle for insects and made many assumptions about their mating habits. Nevertheless, these assumptions led him to the first application of forensic entomology in an estimation of post-mortem interval. His report used forensic entomology as tool to prove his hypothesis on how and when the person had died. The first systematic study in forensic entomology was conducted in 1881 by Hermann Reinhardt, a German medical doctor who played a vital role in the history of forensic entomology. He exhumed many bodies and demonstrated that the development of many different types of insect species could be tied to buried bodies. Reinhardt conducted his first study in East Germany, and collected many phorid flies from this initial study. He also concluded that the development of only some of the insects living with corpses underground were associated with them, since there were 15-year-old beetles who had little direct contact with them. Reinhardt's works and studies were used extensively in further forensic entomology studies. French veterinarian and entomologist Jean-Pierre Megnin published many articles and books on various subjects including the books Faune des Tombeaux and La Faune des Cadavres, which are considered to be among the most important forensic entomology books in history. In his second book he did revolutionary work on the theory of predictable waves, or successions of insects onto corpses. By counting numbers of live and dead mites that developed every 15 days and comparing this with his initial count on the infant, he was able to estimate how long that infant was dead. In this book, he asserted that exposed corpses were subject to 8 successional waves, whereas buried corpses were only subject to 2 waves. Megnin made many great discoveries that helped shed new light on many of the general characteristics of decaying flora and fauna. Megnin's work and study of the larval and adult forms of insect families found in cadavers sparked the interest of future entomologists and encouraged more research in the link between arthropods and the deceased, and thereby helped to establish the scientific discipline of forensic entomology. Urban forensic entomology typically concerns pests infestations in buildings, gardens, or that may be the basis of litigation between private parties and service providers such as landlords or exterminators. Urban forensic entomology studies may also indicate the appropriateness of certain pesticide treatments and may also be used in stored products cases where it can help to determine chain of custody, when all points of possible infestation are examined in order to determine who is at fault. Rove beetles family Staphylinidae are elongate beetles with small elytra and large jaws. Like other beetles inhabiting carrion, they have fast larval development with only 3 larval stages. Criophilus species are common predators of carrion, and since they are large, are a very visible component of the fauna of corpses. Some adult Staphylinidae are early visitors to a corpse, feeding on larvae of all species of fly, including the later predatory fly larvae. They lay their eggs in the corpse, and the emerging larvae are also predators. Some species have a long development time in the egg, and are common only during the later stages of decomposition. Staphylinids can also tear open the pupal cases of flies, to sustain themselves at a corpse for long periods. Beetles family Hysteridae. Adult hysterids are usually shiny beetles which have an introverted head. The carrion-feeding species only become active at night when they enter the maggot-infested part of the corpse to capture and devour their maggot prey. During daylight they hide under the corpse unless it is sufficiently decayed to enable them to hide inside it. They have fast larval development with only 2 larval stages. Among the first beetles to arrive at a corpse are Hysteridae of the genus Saprinus. Saprinus adults feed on both the larvae and pupae of blowflies, although some have a preference for fresh pupae. The adults lay their eggs in the corpse, inhabiting it in the later stages of decay. Carrion beetles family Sylphidae. Adult Sylphidae have an average size of about 12 mm. They are also referred to as burying beetles because they dig and bury small carcasses underground. Both parents tend to their young and exhibit communal breeding. The male carrion beetle's job in care is to provide protection for the breed and carcass from competitors. Ham beetles family Chloridae, carcass beetles family Trogidae, skin/hide beetles family Dermastidae. Hide beetles are important in the final stages of decomposition of a carcass. The adults and larvae feed on the dried skin, tendons, and bone left by fly larvae. Hide beetles are the only beetle with the enzymes necessary for breaking down keratin, a protein component of hair. Scarab beetles family Scarabaeidae. Scarab beetles may be any one of around 30,000 beetle species worldwide that are compact, heavy-bodied, and oval in shape. The flattened plates, which each antenna terminates, are fitted together to form a club. The outer edges of the front legs may also be toothed or scalloped. Scarab beetles range from 0.2 to 4.8 in length. These species are known for being one of the heaviest insect species. Sap beetles family Nidiculidae. Stored product forensic entomology is often used in litigation over insect infestation or contamination of commercially distributed foods. Medico-legal forensic entomology covers evidence gathered through arthropod studies at the scenes of murder, suicide, rape, physical abuse, and contraband trafficking. In murder investigations it deals with which insects' eggs appear, their location on the body and in what order they appear. This can be helpful in determining a post-mortem interval and location of a death in question. Since many insects exhibit a degree of endemism, or have a well-defined phenology, their presence in association with other evidence can demonstrate potential links to times and locations where other events may have occurred. Another area covered by medico-legal forensic entomology is the relatively new field of entomotoxicology. This particular branch involves the utilization of entomological specimens found at a scene in order to test for different drugs that may have possibly played a role in the death of the victim. Song Ci Scorpion flies were the first insects to arrive at a donated human cadaver observed at the Southeast Texas Applied Forensic Science Facility near Huntsville, Texas, and remained on the corpse for 1.5 days, outnumbering flies during that period. The presence of scorpion flies thus indicates that a body must be fresh. Flies are often first on the scene. They prefer a moist corpse for their offspring to feed on. The most significant types of fly include: beetles are generally found on the corpse when it is more decomposed. In drier conditions, the beetles can be replaced by moth flies. Many mites feed on corpses, with macrochials mites common in the early stages of decomposition, while Pteroglyphidae and Orobatidae mites such as Rost rosettes feed on dry skin in the later stages of decomposition. Necrophorus beetles often carry on their bodies the mite Poecilocyrus, which feed on fly eggs. If they arrive at the corpse before any fly eggs hatch into maggots, the first eggs are eaten and maggot development is delayed. This may lead to incorrect PMI estimates. Necrophorus beetles find the ammonia excretions of blowfly maggots toxic, and the Poecilocyrus mites, by keeping the maggot population low, allow Necrophorus to occupy the corpse. Moths, specifically clothes moths family Tinidae, are closely related to butterflies. Most species of moth are nocturnal, but there are crepuscular and diurnal species. Moths feed on mammalian hair during their larval stages and may forage on any hair that remains on a body. They are amongst the final animals contributing to the decomposition of a corpse. Wasps, ants, and bees are not necessarily necrophagous. While some feed on the body, some are also predatory, and eat the insects feeding on the body. Bees and wasps have been seen feeding on the body during the early stages. This may cause problems for murder cases in which larval flies are used to estimate the post-mortem interval, since eggs and larvae on the body may have been consumed prior to the arrival on scene of investigators. Francesco Redi. Bergeret d'Arbois. Rain and humidity levels in the area where the body is found can affect the time for insect development. In most species, large amounts of rain will indirectly cause slower development due to drop in temperature. Light rain or a very humid environment, by acting as an insulator, will permit a greater core temperature within the maggot mass, resulting in faster development. Hermann Reinhardt. Jean-Pierre Megnin. Forensic entomology subfields. Urban forensic entomology. Stored product forensic entomology. M. Lee Goff, a noted and well-respected forensic entomologist, was assigned to a case involving the discovery of a decomposing body found on a boat half a mile from shore. Upon collection of the maggot mass, only one insect, Chrysomia megacephala, was discovered. He concluded that the water barrier accounted for the scarcity of other flies. He also noted that flies will not attempt to trek across large bodies of water unless there is a substantially influential attractant. In addition, the amount of time a maggot mass has been exposed to salt water can affect its development. From the cases Goff observed, he found that if subjected for more than 30 minutes, there was a 2-4-hour developmental delay. Unfortunately, not many more studies have been conducted, and thus a specific amount of delay time is difficult to estimate. Because insects are cold-blooded animals, their rate of development is more or less dependent on ambient temperature. Bodies exposed to large amounts of sunlight will heat up, giving the insects a warmer area to develop, reducing their development time. An experiment conducted by Bernard Greenberg and John Charles Kunich with the use of rabbit carcasses to study accumulation of degree days found that with temperature ranging in the mid-70s to high 80s, the amount of developmental time for maggots was significantly reduced. Medico-legal forensic entomology. In contrast, bodies found in shaded areas will be cooler, and insects will require longer growth periods. In addition, if temperatures reach extreme levels of cold, insects instinctively know to prolong their development time in order to hatch into a more accepting and viable climate in order to increase the chance of survival and reproduction. Hanged bodies can be expected to show their own quantity and variety of flies. Also, the amount of time flies will stay on a hanged body will vary in comparison to one found on the ground. A hanged body is more exposed to air and thus will dry out faster, leaving less food source for the maggots. As the body begins to decompose, a compilation of fluids will leak to the ground. In this area most of the expected fauna can be found. Also, it is more likely that rove beetles and other non-flying insects will be found here instead of directly on the body. Fly maggots, initially deposited on the body, may also be found below. According to Jean-Pierre Megnin's book La Faune des Cadavres, there are 8 distinct faunal successions attracted to a corpse. While most beetles and flies of forensic importance can be found worldwide, a portion of them are limited to a specific range of habitats. It is forensically important to know the geographical distribution of these insects in order to determine information such as post-mortem interval or whether a body has been moved from its original place of death. Calophoridae is arguably the most important family concerning forensic entomology, given that they are the first to arrive on the corpse. The family's habitat ranges into the southern portion of the United States. However, while Chrysomia rufifaces, the hairy maggot blowfly, is part of the family Calophoridae and is widespread, it is not prevalent in the southern California, Arizona, New Mexico, Louisiana, Florida, or Illinois regions. Flesh flies fall under the family Sacrophagidae and generally arrive to a corpse following Calophoridae. However, as previously mentioned, they are capable of flying in the rain. This key advantage enables them to occasionally reach a body before Calophoridae overall, affecting the maggot mass that will be discovered. Flesh flies are globally distributed, including habitats in the United States, Europe, Asia, and the Middle East. Beetles are representative of the order Coleoptera, which accounts for the largest of the insect orders. Beetles are very adaptive and can be found in almost all environments with the exception of Antarctica and high mountainous regions. The most diverse beetle fauna can be found in the tropics. In addition, beetles are less submissive to temperatures. Thus, if a carcass has been found in cold temperatures, the beetle will be prevalent over Calophoridae. Invertebrate types. Various weather conditions in a given amount of time cause certain pests to invade human households. This is because the insects are in search of food, water, and shelter. Damp weather causes reproduction and growth enhancement in many insect types, especially when coupled with warm temperatures. Most pests concerned at this time are ants, spiders, crickets, cockroaches, ladybugs, yellow jackets, hornets, mice, and rats. When conditions are dry, the deprivation of moisture outside drives many pests inside searching for water. While the rainy weather increases the numbers of insects, this dry weather causes pest invasions to increase. The pests most commonly known during dry conditions are scorpions, ants, pillbugs, millipedes, crickets, and spiders. Extreme drought does kill many populations of insects, but also drives surviving insects to invade more often. Cold temperatures outside will cause invasions beginning in the late summer months and early fall. Box elder bugs, cluster flies, ladybugs, and silverfish are noticed some of the most common insects to seek the warm indoors. Many new techniques have been developed and are used in order to more accurately gather evidence, or re-evaluate at old information. The use of these newly developed techniques and evaluations have become relevant in litigation and appeals. Forensic entomology not only uses arthropod biology, but it pulls from other sciences, introducing fields like chemistry and genetics, exploiting their inherent synergy through the use of DNA in forensic entomology. Scorpion flies. Fly larvae and fly eggs are used to aid in the determination of a PMI. In order for the data to be useful, the larvae and eggs must be identified down to a species level to get an accurate estimate for the PMI. There are many techniques currently being developed to differentiate between the various species of forensically important insects. A study in 2007 demonstrates a technique that can use scanning electron microscopy to identify key morphological features of eggs and maggots. Some of the morphological differences that can help identify the different species are the presence/absence of anastomosis, the presence/absence of holes, and the shape and length of the median area. The SEM method provides an array of morphological features for use in identifying fly eggs. However, this method does have some disadvantages. The main disadvantage is that it requires expensive equipment and can take time to identify the species from which the egg originated, so it may not be useful in a field study or to quickly identify a particular egg. The SEM method is effective provided there is ample time and the proper equipment and the particular fly eggs are plentiful. The ability to use these morphological differences gives forensic entomologists a powerful tool that can help with estimating a post-mortem interval, along with other relevant information, such as whether the body has been disturbed post-mortem. Flies. Beetles. Mites. When scanning electron microscopy is not available, a faster, lower-cost technique is potassium permanganate staining. The collected eggs are rinsed with a normal saline solution and placed in a glass petri dish. The eggs are soaked in a 1% potassium permanganate solution for 1 minute and then dehydrated and mounted onto a slide for observation. These slides can be used with any light microscope with a calibrated eyepiece to compare various morphological features. The most important and useful features for identifying eggs are the size, length, and width of the plastron, as well as the morphology of the plastron in the area around the micropyle. The various measurements and observations when compared to standards for forensically important species are used to determine the species of the egg. In 2001, a method was devised by Jeffrey Wells and Felix Sperling to use mitochondrial DNA to differentiate between different species of the subfamily Chrysominae. This is particularly useful when working to determine the identity of specimens that do not have distinctive morphological characteristics at certain life stages. A valuable tool that is becoming very common in the training of forensic entomologists is the use of mock crime scenes using pig carcasses. The pig carcass represents a human body and can be used to illustrate various environmental effects on both arthropod succession and the estimate of the post-mortem interval. Although physical characteristics and sizes at various instars have been used to estimate fly age, a more recent study has been conducted to determine the age of an egg based on the expression of particular genes. This is particularly useful in determining developmental stages that are not evidenced by change in size, such as the egg or pupa, and where only a general time interval can be estimated based on the duration of the particular developmental stage. This is done by breaking the stages down into smaller units separated by predictable change in gene expression. 3 genes were measured in an experiment with Drosophila melanogaster, bicoid, slalom, and chitin synthase. These 3 genes were used because they are likely to be in varied levels during different times of the egg development process. These genes all share a linear relationship in regards to age of the egg, that is, the older the egg is, the more of the particular gene is expressed. However, all of the genes are expressed in varying amounts. Different genes on different loci would need to be selected for another fly species. The genes' expressions are mapped in a control sample to formulate a developmental chart of the gene expression at certain time intervals. This chart can then be compared to the measured values of gene expression to accurately predict the age of an egg to within 2 hours with a high confidence level. Even though this technique can be used to estimate the age of an egg, the feasibility and legal acceptance of this must be considered for it to be a widely utilized forensic technique. One benefit of this would be that it is like other DNA-based techniques, so most labs would be equipped to conduct similar experiments without requiring new capital investment. This style of age determination is in the process of being used to more accurately find the age of the instars and pupa. However, it is much more complicated, as there are more genes being expressed during these stages. The hope is that with this and other similar techniques a more accurate PMI can be obtained. A preliminary investigation of insect colonization and succession on remains in New Zealand revealed the following results on decay and insect colonization. This environment had a daily average maximum temperature of 19.4 degrees Celsius and a daily minimum temperature of 11.1 degrees Celsius. The average rainfall for the first 3 weeks in this environment was 3.0 mm/day. Around days 1745, the body began to start active decay. During this stage, the insect succession started with Calliphora stygia, which lasted until day 27. The larvae of Chrysomia rufifacies were present between the day 13 and day 47. The H. rostrata, larvae of Lucilia sericata, Psychotidae family, and Sylvicola were found to occur relatively late in the body's decay. This environment had an average daily maximum temperature of 21.4 degrees Celsius and minimum of 13.5 degrees Celsius. The daily average rainfall was recorded as 1.4 mm/day for the first 3 weeks. The post-decay time interval, beginning at day 6 after death and ending around day 15 after death, is greatly reduced from the average post-decay time, due to the high average temperature of this environment. Insects obtained late in the post-active stage include the Calliphora quadrimaculata, adults Phaeroceridae, Psychotidae, and Pyophilidae. This environment had recorded daily average maximum and minimum temperatures were 18.0 degrees Celsius and 13.0 degrees Celsius, respectively. The average rainfall in this habitat was recorded at 0.4 mm/day. After the bloat stage, which lasted until day 7 after death, post-active decay began around day 14. In this habitat, the H. rostrata, adult Phoridae, Sylvicola larvae, and adult were the predominant species remaining on the body during the pre-skeletonization stages. Throughout its history, the study of forensic entomology has not remained an esoteric science reserved only for entomologists and forensic scientists. Early 20th century popular scientific literature began to pique a broader interest in entomology. The very popular 10-volume book series, Alfred Breham's Tierleben, expounded on many zoological topics, including arthropods. The accessible writing style of French entomologist Jean-Henri Fabre was also instrumental in the popularization of entomology. His collection of writings Souvenirs entomologiques, written during the last half of the 19th century, is especially useful because of the meticulous attention to detail to the observed insects' behaviors and life cycles. The real impetus behind the modern cultural fascination with solving crime using entomological evidence can be traced back to the works Fonds des Tombeaux and Les Fonds des Cadavres by French veterinarian and entomologist Jean-Pierre Megnin. These works made the concept of the process of insect ecological succession on a corpse understandable and interesting to an ordinary reader in a way that no other previous scientific work had done. It was after the publication of Megnin's work that the studies of forensic science and entomology became an established part of Western popular culture, which in turn inspired other scientists to continue and expand upon his research. Moths, wasps, ants, and bees. Factors: moisture levels, submerged corpses, sun exposure, air exposure, geography, weather, modern techniques, scanning electron microscopy, potassium permanganate staining, mitochondrial DNA, mock crime scenes, gene expression studies, insect activity case study, open field habitat, coastal sand dune habitat, native bush habitat. In literature. Notes.